Just joining, so I'll just wait for 1 more minute before I begin. Okay, so we're there. So today we'll begin to study biotechnology. Okay. Yes. Let all of them join. Very well. I think everyone is joined. Good evening, Sumaira.
Good evening, sir.
And what? Good evening. Okay, so a new chapter will begin today, biotechnology. In this chapter, we'll be dealing about the principles, basically the fundamentals. It's like a introduction to biotechnology, the field of biotechnology. And you must be knowing that biotechnology has both the aspects. So you must be knowing that there's a degree in engineering called BTech Biotechnology. There's also a degree in science, like which is not engineering, but it's the same. It's BSc Biotechnology. You know that?
Yes, sir.
Yeah. So basically, it's something where we are trying to mix up technology, like engineers and biologists. Okay. So as the term suggests, it has two components: bio and technology. So what do you think the word biotechnology means, or what all things should come under the umbrella of biotechnology to begin with? So if it is if I use the word let's say radio technology, or I use the word telecom technology. So you know, something with radio technology has something to do with radio waves, right? Sound frequencies. Biotechnology, the word bio means life, right? Or I'll say living systems. Correct? What does
Yes,
the
sir.
word technology means?
Machines.
Machines, yes. Machines are a product of technological advancement, but technology. The word logy means, always see in science, it's very simple. Break the word biology. If I remove the techno word from the middle and I say biology, you will say study of life or living systems. Simple, right? So when I say technology, you don't have to look at it as one word. Logy means study, always, any field: physiology, cosmology, neurology, biology. What does the word techno means? Techniques. Correct? So what does the word biotechnology to be combined means?
Techniques to study living systems.
Yes, kind of, but you forgot one thing. The living first comes. So it's the field which uses living systems, okay, to develop techniques using the living systems which we can study, and we can produce some useful products for mankind. Okay. So it's the integration of natural sciences along with living systems, cells, proteins, enzymes within the living systems to make goods, products, or services for the mankind. So write down: biotechnology is the integration of natural sciences and or living systems is the integration of natural sciences or living systems with
Sir, can you repeat again?
biotechnology is the integration of natural sciences and living systems with their molecular components and techniques to develop useful products and services. And techniques to develop useful products and services. So going by this broader definition, in other words, write down one more sentence. In other words, biotechnology deals with techniques of using living system and their components. Techniques of using living systems and their components to produce
The living systems and their
components like enzymes, proteins, toxins, anything that they produce. Living systems and their components to produce useful products or services for humans. Useful products or services for humans, majorly for humans. Yes. We can also use biotechnology for other animals. It's not like it's just human centric, but for now we'll use the word humans, okay? Because that's how it started. We did not want it to develop a new technique so that it can help our dogs. We first did it for us and we realized, oh, this way we can also help other organisms. So we first started making vaccines for humans. Then we realized that certain vaccines can also be made for our animals, pets, because they also get certain diseases. So that's why I'm using the word humans here, okay? We use the same techniques for plants, agricultural plants, and our pets as well. Make sense, everyone?
yes, sir.
Okay. Now, what all things come under biotechnology? So going through this broader definition, even making curd or a bread or wine, because we are using some microbes there, right? We are using lactobacillus to make curd that we studied in the last chapter. So what is the technique? There is no technique there. The technique is just put some inoculum into the milk and it will make the milk curd. For the bread, just put dough with yeast, store it for a long time overnight at a particular temperature, and it will make dough into bread, fluffy. So this can also be considered as a biotechnological activity. But since we are going to read about techniques and technologies that has helped us to do things on a large scale, which is called on a factory scale for large productions. We will not discuss curd making, bread or wine making at homes in this process, okay? But if you are thinking along those lines, you are not incorrect. Basically, that's also biotechnology. But what we will be talking about is, for example, let's say in vitro fertilization. We have studied this in the chapter reproduction. So what do we do in vitro fertilization? Anyone quickly? What is in vitro fertilization? IVF. Mamona.
So,
Yeah. Or yes. Go on, Safi.
so we take the gametes from male and females and fertilize it outside the human body.
Correct. So what living system we are using here? We are using gametes, which are living cells, sperms and ovum, and we keep them living even outside the body. We make them fertilize outside the body with the help of technology and techniques. Then we implant it back into the body. So we are doing this to make a service or a process that is useful for humankind. This was not possible without sophisticated machines and technology. But making a curd is possible without a machine, right? But basically, in both the cases, the fundamental thing is we are utilizing living systems to do something that was not happening on its own or was difficult to happen. So in vitro fertilization is a good example of a biotechnological process. Anything else on the basis, on the same lines? Anyone can suggest anything else which comes under biotechnology? People, recently we saw a great product of biotechnology which saved millions of lives. Making RNA or DNA vaccines.
Vaccine.
Yes, making RNA vaccines or DNA vaccines. So we have made vaccines against COVID, which are RNA vaccines because COVID virus have a RNA genome. So that could not have been possible without biotechnological tools, correct? Anything else?
Answer is bioengineering. That BT cotton, what we studied last time.
Perfect. Bioengineering, or let me call it genetic engineering, right? Genetic engineering comes under, or you must have heard of a word called gene therapy. Have you heard of gene therapy, where we treat certain gene anomalies, sorry, gene therapy. All these we can do because we have biotechnological tools to do that. So you're right, correct. BT cotton is a very, very good example. What we did in BT cotton, I think we studied BT cotton in the last class only, correct? Where we use microbes in human welfare. So Safila, would you like to tell what is a BT cotton?
So BT is a species that is like
What does BC stands for? Sorry, BT stands for. Anyone, what does BT stands for? See, this we just did in the last class, I guess.
the Bacillus thuringiensis.
Bacillus thuringiensis, right?
Yes.
Correct. It's a bacteria that produces a toxin that kills the insects. So we put that gene into the plant itself, in the cotton plant. Now, if the insects attack the plants and eats it, the insect is dead. So that's genetic engineering, again, a biotechnological application. So today, let's start with principles of biotechnology. So what are the basic principles? So by principles, I mean, what are the core techniques? So we can do multiple things. We can apply biotechnology in hundreds of different fields, in agriculture, like BT cotton, in healthcare, like making vaccines, in reproductive techniques by in vitro fertilization, in treating diseases, again, healthcare, like gene therapy. We can do multiple things, okay? We can also make high yielding varieties of crops. We can make different breeds of cats and dogs sometimes even using this thing. But point is, what are the core techniques that have led to the birth of biotechnology? So the core techniques are just two. Okay, so write down. There are two core techniques in biotechnology, in the field of biotechnology. In the field of biotechnology. Whenever you want to engineer, alter, modify, do anything with a living system, what is the first target that you can edit, modify, or work with? You want to change some functions of an organism, you want to give any function to an organism, you want to make human insulin into bacterial cells, anything that you want to do in biology, any kind of bioengineering, or any kind of using the living system to produce something, what can we do? We can tweak the genome, the genetic material. That's all what we can do, correct? Every living system has a library where if you can access that library, you will get to know each and everything about that living system, each and every property of its phenotype, the kind of enzyme it produces, and that library is called the DNA sitting inside the nucleus, which is the library room. So the first technique, which is common in all biotechnological processes, is genetic engineering. Genetic engineering is a core technique. So if you are a genetic engineer, you're not going to make bridges or towers or architectures. You're going to make DNA and RNA. Sounds like a good engineering job, right? And believe me, it requires the same concepts of precision, accuracy, timing, etc. So genetic engineering is first. Well, let's define it. Write down. Genetic engineering involves all the techniques to alter slash modify, alter or modify the chemical properties of genetic material. The chemical properties of the genetic material. And what is the genetic material? DNA or RNA, okay? And why are we doing this modification in the chemistry of genetic material? Continue writing. These modified or this modified genetic material, this modified genetic material this modified genetic material can be introduced into a host organism to change the phenotype of the host organism. That can be any phenotype. To change the phenotype of the host organism. So this is what we do basically. First core principle that stays always. Whether we are trying to make a new protein through genetic engineering in bacteria, or we are trying to do anything else in any other organism. Second core technique is bioprocess engineering. So either we engineer the genetic material, or we engineer something that helps with this process. Bioprocess engineering. Now, to do anything, like for example, to do in vitro fertilization, what do we need?
Gametes from the mother or father.
Just that?
The tools and sterile condition for the fertilization to take place.
Yes, we need a lot of supportive environment also, right? Because we are taking just the gametes out of the body, does not mean if I take the gamete outside the body and just put it anywhere in, let's say, one glass of water, they will fertilize, start making embryo, then I can again take that embryo back in a syringe and put it back into the body. We have to make sure that there is no contamination of any kind when we are doing these genetic engineerings. So we have to maintain. So even maintenance of the sterile condition or the ambience in any chemical engineering process, okay, or to choose specifically a condition in which only one type of cells will survive and the other types will not survive, okay, or basically to make things like antibiotics, vaccines, enzymes, everything in a very large quantities, we need a full process engineering as well. So we need genetic engineering, and then we need the bioprocess engineering. Now write down. The bioprocess engineering includes maintenance of sterile conditions. What do we mean by sterile, everyone? Everyone knows sterile? Microbes free. Microbial contamination or microbial growth free environment is called a sterile environment. The word sterile is also used for organisms that cannot reproduce. But here it's microbial contamination free environment. Maintenance of sterile conditions to facilitate growth of only the desired microbe or living cell. Only the desired microbe or living cell in large quantities. In large quantities. Okay. So first thing is to tweak the DNA or RNA. That's done. Now, whatever tweaking you did, you want to see its effect. Let's say you tweaked the DNA or RNA to make an enzyme. Now you want to produce that enzyme in bacterial cells. So you will also do bioprocess engineering to make sterile environment, all the equipments, the process, everything to grow these bacterial cells, and only these bacterial cells. You also do not want any other contamination. You want to be 100% sure that whatever you are growing in that culture plate, or in that vessel is just your bacteria containing your desired gene or DNA or RNA that you have modified. So that all comes under bioprocess engineering. Okay, write down. Next point, bioprocess engineering leads to large scale manufacturing of biotechnological products. Large scale engineering of biotechnological products. For example, antibiotics, vaccines, enzymes, etc. Supplements also. Vaccines, enzymes, supplements, medicinal drugs, etc. Okay. Is it clear, everyone? Are the two core components or core techniques of biotechnology clear to you?
Yes, sir.
Okay. Very good. So now let's try to go into some depth and read some of it. For example, how are we actually going to do so in detail whatever I told you above, like making of antibiotics, expressing one gene from one organism into another organism where that gene was not present. For example, when we were discussing in previous chapters, I told you that you can make human insulin into a yeast or a bacterial cell. We studied about YAC and BAC when we were doing the human genome project, right? Or human fingerprinting, DNA fingerprinting, right? So there are bacterial artificial chromosome and yeast artificial chromosome, correct? All these are a part of biotechnological tools. So we are going to go into the details of it. Now, what we actually do is, what is the, remember what was the advantage of sexual reproduction over asexual reproduction?
Variation.
Perfect. It was variation. And how was that variation coming, Sahel?
Different coding in the DNA.
Different coding in the DNA. But different coding, but what do you mean by different coding?
Like coding for each organism is different, right? Like,
Correct.
yeah.
Yes. So genetic variation is
Yeah.
there between different organisms because they are like the coding, the sequence is different. You mean to say.
Sequence of the coding is different.
Yes. So the code is same only. The
Yeah,
different genes have different sequences of ATGC only. There are four codes only. But I'm talking about when I say sexual reproduction, we are not talking about mouse and human. We are talking about human and human. Because mouse and human cannot have sexual reproduction, right? Or for that matter, any kind of reproduction, because they are not belonging to one species. Even even among the individuals of the same species, there is variation, right? Genetic variation. Some have blue eyes, red hair, blonde hair, black hair, curly hair, straight hair, dry skin, oily skin, good immunity, bad immunity. Where is that coming from? That is the variation that I'm talking about, which is coming from sexual reproduction. Now, how are these variations coming from? Why is someone tall and the other one is short? Why is someone having a darker skin, other having a fairer skin? Why someone is more tolerant, have more tolerance towards some specific pathogen or a foreign particle, but other person does not have that much of tolerance? Why do parents, let's say parents had, both of the parents had black hair, but the baby had brownish hair? Both the parents had straight hair, but the baby had a curly hair. Where did that variation came from? Sexual reproduction, of course, but how in sexual reproduction? You cannot forget these things. These are basics. Come on, go back to the chapter and see what was responsible for
The genes for curly hairs might be passed on from the parents, from their parents.
But parents had all straight hair.
But somewhere
There
in the
might
ancestry.
Yes, yes. I'll go first to Saifullah and then Sahel. Yes.
say the genes might be present in them, just not expressed yet.
So you mean that that gene was recessive gene in parent?
Yes.
Then how did it expressed in the kid? Why is not, why there also it has to be recessive only, no? Recessive gene cannot be a dominant gene when it passes on. It cannot change. It will still be a recessive gene only.
There might be like from
It can be expressed right in the next generation because once
Yes.
every generation a recessive gene is expressed.
How? What's happening?
genes might be coming from both the parents. So
The recessive allele copy must be coming from both the parents, right?
yes.
Yes. So the only copy left in the kid is both the recessive copies. So of course they will express. That is one way. Very good. Now remember, we also talked about more than one genes. Let's say the parent had curly hair with fair skin, and the other parent had straight hair with a dark skin. But the kid had a curly hair with a dark skin, which was not a combination present in any of the parents. How is that possible?
Sir, it may have been present in the grandfather or the previous generation.
So how does it matter to the generation? For them, what matters is what is coming from the parents. So if one parent had a curly hair with a fair skin, and if his genes expressed, then the kid must be curly haired and fair skinned. If the other mother had straight hair with a dark skin, and if that parent's gene expressed, then the kid should have dark straight hair and dark skin. But why, how can a kid have a fair skin with a curly hair, which is a combination not present in any of the parents? And let's say it was not also present in any of the grandparents as well.
In complete dominance, then.
Nothing to do with dominance. I told you about something called recombination, genetic recombination, where physically the chromosomes get broken and a piece of it is exchanged between two chromosomes at a region where they cross over. That's called chiasmata formation during meiosis. I told you this. I draw some fancy colored chromosomes. We took 3 genes, capital T, capital R, and capital Y. On the other, it was small t, small r, and small y. Then some were called recombinant populations. Forgot everything, right? People, recombination. Please go back and revise, okay? So recombination is one reason apart from mutations. So mutations are random and sudden and they do not have a direction. So we'll not consider mutations here, but genetic recombination we will consider. So genetic recombination is where variations come from unique combinations from the parents. Okay, so this can be beneficial to the organism. These recombinations can be beneficial. In asexual reproduction, the genetic information is preserved as it is. So there is no variation unless and until we are not taking into account mutations here, let's say. So if mutation, because mutations are it can happen anywhere, both in asexual reproducing organisms and or it can happen in sexually reproducing organisms. It has nothing to do with the process of reproduction itself. It can happen at any given point of time, anywhere. Okay, so for now, we will consider that genetic recombination leads to variations which is present in sexual reproduction and not present in asexual reproduction. Now, on the same concept, we started hybridizing or producing plants and animals with different breeds. So let's say we also discussed about animal breeding when we talked about cats and dogs, right? Yes or no? Do you remember breeds of cats and dogs, artificial selection, natural selection?
Yes, sir.
Yes. So how could we manage to make a dog with some characteristics of, let's say, a German Shepherd and some characteristic features of Golden Retriever or Dalmatian for that matter? By crossbreeding them, right?
Sir.
Yes. Yes. Because we want some properties from one and some properties from other. Now imagine the same thing we do in a engineered way, like in the same technique we use in genetic engineering, and we create recombinant DNA with the use of techniques like gene cloning and gene transfer. So we are not just relying on how it naturally happens, because naturally, suppose you want a specific characteristic of one organism and the other specific characteristic of other organisms expressed together. So for that, we have to let them mate sexually and produce enough number of progeny, so that it's not like the first time we will make them mate and all the progenies we will get will have the combination that we want. There will be many without the combination also. Yes or no? Do you understand, people?
Yes, sir.
Yeah. So we just want to be 100% sure of that only those characters or genes, or only those things are expressed. So for that, what we do is we artificially create recombinant DNA. Okay. Now, what happens to a DNA? Let's say, before I go into the details, let me ask you, what happens to a DNA inside a cell? If I put any DNA inside any of your cell, what will happen to that DNA?
It will be destroyed.
Why?
Because, sir, it is a outside organism that is hostile to the cell, cytoplasm of the cell.
No, but I'm talking about the molecule DNA. If it is an outside cell, if it's a pathogenic cell, you are right. If it's a bacteria, of course, our body will recognize that it's not my cell, it's a different behaving bacteria. It's a bacterial cell. But if I just take the DNA from that bacteria and just that DNA molecule I inject into your cell, what will happen?
It may create a recombination and into another organism.
What? How will it make a recombination?
So not a recombination, but it may bind with the DNA of our DNA.
Yes, perfect. It can get incorporated into the host DNA, correct? And this is exactly what happens during infection of HIV/AIDS. I taught you HIV/AIDS, right?
Retrovirus RNA recombination.
So retroviruses release the mRNA because their genome is a RNA genome. When it comes into the cell, what happens? It does reverse transcription to become a DNA, correct? Remember that?
Yes, sir.
So a piece of DNA of a host cell can get incorporated by the or in the host genome. Incorporated in the host genome. And whatever that piece of DNA carries, let's say that piece of DNA carries a gene. So what will happen? That gene will start expressing in the host cell now. Yes? Yes or no, people? Are you understanding what I'm saying?
Yes, sir.
Or all of you are sleeping? Because anyways, I cannot see you. None of you are speaking also. So it becomes difficult for me to understand if you're getting it. So a piece of DNA in a cell can get incorporated in the host genome and can give the same abilities to the host that the DNA itself carries. If it carries a gene, let's say a bacteria took up a host, bacteria being a host, took up a DNA fragment just lying around, and that DNA fragment had a gene that can help you survive. That gene produces a protein that can protect you from high temperature, let's say, or can help you survive better in high temperature. So that gene or that piece of DNA will start giving that bacterial cell the ability to survive better in hot conditions. Yes or no?
Yes, sir.
Sir.
Yeah. So don't you think this way we can actually transfer characteristics from one organism to another? We can transfer genes, let's say. By this way, we can transfer genes, yes or no? Simple.
Yes, sir.
And by genes, we can also transfer ability, like characteristics. Gene gives rise to characters. We can clone a gene in one organism. We can make multiple copies of a gene in one organism and then can purify it. But how do we do all these things? Is the question. Write down. To create a recombinant DNA, we techniques like gene cloning and gene transfer are used. So simply gene cloning. What do we mean by gene cloning? People, gene cloning? Simple, 2 words, gene and cloning.
Making copies of gene.
Making copies of gene without changing it in any small amount, right? So how can
Yes.
you do, what is, how will you define it? How can we make copies of gene? Go to central dogma, it will give you the answer. The first step of the central dogma is the answer.
DNA to RNA, RNA to protein.
This is the gene expression you're talking about. How is it cloning then? Where are you making copies here? You are making DNA to RNA, RNA to protein, protein get expressed, work done. The first step, replication, is nothing but gene cloning only, right? In a cell, the DNA replicates to make more DNA. The gene can replicate to make new gene, the same gene, another copy of the same gene. In a haploid cell, there is one copy of each gene. In a diploid, there are 2 copies of each gene, right? So when a DNA is replicating, when the cell is dividing, it is making more copies of itself, isn't it?
Yes, sir.
Isn't it gene cloning only? But if we want to do it on a large scale, what we can do, write down gene cloning is the method of preparing or producing Gene cloning is the method of producing multiple identical copies of the gene sized pieces of DNA. Gene sized multiple identical copies of the gene sized pieces of DNA, or simply multiple identical copies of the DNA containing a gene, containing a desired gene, any gene that you want. So you don't have to replicate the whole DNA over and over again. You can just take the fragment of the DNA that you are interested in making copies of, because that contains your gene, and you can do it through DNA replication. Now, inside our cell, the DNA only replicates when the cell has to divide. Correct? Yes or no?
Yes, sir.
Yes, sir.
How can we do it artificially? Any idea how can we do it artificially? We can do it artificially by using something known as plasmid. You know about plasmid? We can ask a bacterial cell yes, Sahel, okay, it's completely fine. You can see it through the recording, okay?
Thanks, sir.
Yeah.
Bye.
So if you can make a bacterial cell make multiple copies of the gene of your interest, then it's simply gene cloning, right? Because we know what we know about the bacterial cells. A bacterial cell copies its genome every 20 minutes. Let's say E. coli divides after every 20-25 minutes. Yes or no?
Yes, sir.
So if we want to make millions of copies of our gene, we can just make, even if bacteria is making one copy of our gene every time it divides, in a few days we can get millions of copies of our gene, right? Which is not possible in our own cell when our cells replicate. Don't you think it's a biotechnological tool to clone the gene? Yes. And what is gene transfer also? Second word was gene transfer, gene cloning and gene transfer. So we'll talk about gene cloning again. But what is gene transfer? Gene transfer, simple. You transfer any gene to any other organism and see what happens when that gene gets expressed in that organism, into the target organism. Okay.
So gene transfer is a process of transferring the recombinant DNA to the desired target organism.
Yes. So what is this recombinant DNA? That recombinant DNA is the DNA that contains our gene of interest. And let's say we want it to be transferred into some other organism. So the gene that we were cloning first needs to get transferred from our cell to the bacterial cell. And that process is called gene transfer. And it actually helps us, the process of gene transferring, which you said through a recombinant DNA technology, we can only take the gene that we want and transfer it into any other organism without worrying about any undesirable genes getting into the target organism. So whatever we want, we can transfer that very, very, what you can say, precisely. Make sense, everyone? Now, if a DNA just enters the cell but fails to incorporate itself into the host genome, that piece of DNA will be degraded by the cell. Because any DNA or RNA, both these are nucleic acids, inside the cell, they are not very, very stable for very long. DNA is stable than RNA, but still, if it's a alien piece of DNA just floating around, it needs to get incorporated. Now, what do you need for it to get incorporated? That's also a question. Okay, so that it can multiply. So it will only be stable and protected if it becomes the part of the chromosome in the host cell, and then it can replicate. Right? Because I told you that in chromosomes of every organism, there is something called origin of replication. Remember, I told you that if there is a cell, this is a cell. Inside the cell, now the nucleus has opened. Let's say it's the prophase, and it's going toward the metaphase. So in prophase, the nucleus disappears first, and your DNA is naked, and it starts forming a chromosome. And you put some foreign DNA. Okay. Now, in this chromosome, when replication happens, it's not like replication starts from one end. So if I take this out, this is one piece of the DNA, let's say. When it replicates, it's not like it starts from one end and goes all the way to the other end, and one DNA strand is replicated. Replication happens from multiple different sites simultaneously, right? And these are called origin of replication. This you must have studied in the cell chapter, cell division chapter. Have you studied this? Do you remember this? There are multiple origins of replications in a DNA, in a chromosome. So write down, what is origin of replication? Origin of replication is the region of the DNA sequence region of the DNA that is responsible for initiating the process of replication. Initiating the process of replication. Okay. So now we know two components. To successfully clone any gene, we have to do a successful gene transfer, where we make sure that our gene gets into the cell, and there are two ways. Either it gets into the chromosome of that host, because if it does not incorporate itself with the host, it may not be able to replicate, because only the host chromosome have certain sequences which are called replication site, origin of replication sites. So our gene that we have cut off is just the gene. It has nothing else. No origin of replication is there. So that's why making it incorporate into the genome becomes very important. Is there any other way that you can think of? If let's say we do not want it to get incorporated in the genome and still want to make copies of the gene. Can we do anything else? Because there is no concept of a nucleus in a bacteria, no? People, I think you all have slept, right? Or are you listening? If you're listening, no one is even either saying yes, you're understanding.
Yes, sir.
Yes. Is there a concept of nucleus in bacteria?
Yes, sir. There is.
Yes.
No, there is no concept of nucleus in bacteria. That's where you are sleeping. Bacteria is a prokaryote. There is no nucleus in a bacteria. The DNA is lying naked in a dense region called nucleoid, but there is no membrane. Correct?
Yes, sir.
Yeah. So if there is no concept of nucleus in a bacteria, replication is just happening in the cytoplasm only, right? And I told you that replication is clubbed with, transcription is clubbed with translation.
Yes, sir.
And replication is also happening just in the cytoplasm, in the nuclear region. So if I cannot make my gene, or if I do not want purposefully to make my gene get incorporated in the bacterial genome, what else can I do? I know two things now. One, it should not be lying around simply, and it needs origin of replication to make copies. What can I do? I can provide my gene, desired gene, with its own origin of replication sequence. Yes? If my gene sequence have its own origin of replication sequence, when it will go into the bacterial cell, it will start getting replicated, right?
Yes, sir.
Simple. But then again, it's a DNA fragment. It can still be unstable and can be degraded by other factors. So for this, we make artificial recombinant DNA molecules or artificial chromosomes. BAC and YAC, Human Genome Project, bacterial artificial chromosome and yeast artificial chromosome. What are these? These are circular pieces of DNA. Okay. Because they are circular, they don't have any loose ends from where degradation can happen very easily. Make sense, everyone?
Yes, sir.
It's a circular piece of DNA. And what happens is, there are some things called plasmids. In the cell chapter in your junior classes, you have studied that bacteria have plasmids. They are called extra chromosomal circular DNA. You know about plasmid, everyone? Do I have to tell you this? So a bacteria, let's make a bacterial cell here to make you understand. So apart from, let's say this is a E. coli. So it will have a single chromosome, which is its genetic material, and most of the time their chromosomes are also circular. Apart from these, they have small plasmids, right? So this is the genetic material, which is DNA, and these are plasmids, which are separate. Plasmids are extra chromosomal DNA present in bacteria. Okay. And the property of plasmids is that they can replicate independent of the genetic material. So they are autonomously replicating. Autonomously replicating circular DNA. Now, anyone's curiosity got clicked Oh, this is something amazing, right? What can we do with this? Now, we do not have to necessarily, whatever gene we want to clone, we do not have to depend now on the replicating of the cell of the bacteria itself and its genetic material. We found that there can be extra chromosomal DNA in the bacteria that autonomously keeps replicating even when the cell is not dividing itself. Inside that cell also it replicates autonomously on its own. So does it click something in your head? Do you think it's useful? Come on, people. Don't make this class so boring.
Can you repeat the question, please?
The question is on the screen. What do you see on the screen? What is this?
Plasmids,
What is this?
genetic material, E. coli.
E. coli, this is the genetic material. What are these two?
Plasmids,
What are plasmids?
extra chromosomal DNA,
Yes, which has the property of
autosomally replicating cellular DNA.
sorry, someone said something else. Property of
encoding
encoding genetic information. Yes, but that every DNA has. This is plasmid, which is extra chromosomal DNA. This also encodes information. This also encodes information. How are plastids different from the chromosomal genetic material? A bacteria, by the way, has just one chromosome. Every bacteria has just one chromosome. The other important property is that it can autonomously on its own replicate in the bacteria. So now, do you think if instead of incorporating my interested gene of desire, let's say I want to make insulin for humans to help diabetic patients. Now, where do I make insulin? Or to first make insulin, I have to make multiple copies of the gene of insulin, right? Yes, no, people.
Yes, sir.
So it comes down to the point where I want to clone any gene. I want to clone my gene of interest so that I can do other things later on. So to clone my gene, now can I just put it in a plasmid where the plasmid acts like a vector? I put my gene. So if this is a plasmid, let's say if I make a bigger, I take this frame and zoom it here. So this is my plasmid. And let's say my gene, I can insert my gene of interest between the two genes like here. From this region to this region, this red mark is my gene of interest in this plasmid. So when this plasmid will replicate, my gene will also replicate, right? And all it needs to replicate is a origin of replication. And if plasmids are already replicating, they must be having origin of replications. And if they are replicating autonomously multiple times, they must be having more origin of replication than normal chromosomes have. Correct?
Yes, sir.
So I can just put my gene of interest in this plasmid, send this plasmid into the DNA. And how to send plasmid into the DNA, that is something that we will cover in the next class. But know that there are techniques by which we can send plasmids into the bacterial, sorry, plasmid not into the DNA, plasmid into the bacterial cell. So there are techniques by which we can make bacterial cells competent cells. So this is your homework for today. You have to go and read what are competent bacterial cells. It's very simple to do. We used to do it routinely in the lab. It's a standard protocol. The logic behind it is simple. I'm not saying it's simple to do like by hand, but the logic is simple. It's straightforward. We have to make because these plasmids are very, very small as compared to the whole genome of the bacteria, which again is very small as compared to the human genome, of course. But if they are very small, small circular pieces of DNA, they can be taken up by the bacteria. We will talk about it how later. But now I got a better tool to use, correct? I can insert my desired gene in a plasmid, and that plasmid will act as a plasmid vector for my gene. I can send it into the bacteria, and I can associate my desired gene with a high copy number origin of replication. So the copy number is decided by the plasmid sequence itself. So multiple copies of my gene will be produced from the bacterial population. Does this make sense, everyone?
Yes, sir.
Yeah. Okay, so now let's write everything down that we have discussed till now so that you can consolidate in your memory. So write down. In
Is there any subheading you have to write?
yes, we are just writing, write down cloning of our gene of interest. Cloning of the gene of interest, whatever gene you are interested in cloning. This is the heading. Write down. To clone any specific gene of interest, to clone any specific gene of interest, our gene sequence has to be incorporated with the chromosome of the host cell. Chromosome of the host cell, because only the chromosome has the ability to replicate. Only a chromosome has the ability to replicate due to the presence of specific DNA sequences called the origin of replication. Due to specific DNA sequences called the origin of replication. Next point, this origin of replication sequence can initiate replication. Only this sequence can initiate replication. Okay. So this is the first thing that we have to do. which means the other thing that we can do is write on the next point. Therefore, any alien DNA, therefore, any alien DNA which is linked with the origin of replication sequence, any alien DNA which is linked with the origin of replication sequence can replicate and multiply itself in the host organism, can replicate and multiply itself in the host organism. This process is called cloning of the gene of interest. This process is called cloning of the gene of interest. So whatever we discussed, I'm just making you write that all so that you can write it in exam. So you understood what is cloning of a gene of interest?
Yes, sir.
Yes, sir.
So this is the traditional thing. This is the knowledge. We know that we have to, it's mandatory that we have to link it with origin of replication. Now scientists started thinking, okay, so let's do that. But like I asked, can we also do something where our gene of interest can have its own origin of replication and can still stay stable, not get degraded, also not get incorporated in the main genome, and we can make multiple copies? Because that will make our lives easier. That way we also don't have to just depend on the bacterial replication, because that has a rate, that has a fixed rate. We can make things even faster. So if bacteria replicates in 4 hours to make 10,000 copies, it's not like our gene will just have 10,000 copies. We can have more than 10,000 copies just by having our own origin of replication. So that the answer is yes, we can do that. We can make recombinant DNA and we can link our gene of interest with a plasmid in the bacteria, because we know that bacteria on a regular basis can autonomously replicate plasmids in its cell. Now, plasmids have this autonomously replicating behavior. So the first experiment that was done to see if we can artificially construct a recombinant DNA molecule or not was done. So how was it done? So write on this part. Construction of an artificial recombinant DNA molecule. This part is clear to you, everyone?
Yes, sir.
Cloning
Yes,
of
sir.
gene of interest. So cloning part is clear. Cloning we can do for that matter in any cell, but then we have to depend on the cell's chromosomes origin of replication. Correct? So let's go one step ahead. Write down construction of the recombinant DNA, or construction of an artificial recombinant DNA molecule. Now this is genetic engineering, right? We are constructing a DNA with our gene of interest and with origin of replication. So what they did, they figured out that there is plasmid. So the bacteria that was taken was Salmonella typhimurium. You have studied this, right? What does Salmonella typhimurium does?
Typhoid.
Yes. So which means it's a pathogenic strain, correct?
Yes, sir.
But this pathogenic strain can be of use if we, so of course, when we are doing experiments with pathogenic strains, proper precautions are taken. A full body mask is worn and you wear mask and use gloves, and you do not let this bacteria contaminate or go into your lungs or on your hands or through the saliva in your body. But what we do is Salmonella typhimurium have a native plasmid. So it has a native plasmid. And plasmid, I've already told you, right? You have written what is plasmid? Autonomously replicating things. Yes. So what we did, we have to check whether this technique is working or not. How can you check that? Any idea? If I put my own gene, I will not be able to check whether this plasmid is replicating my gene. And let's say I cannot, can I put a gene of for insulin or something else? I may not be able to check it actually. So to check, we need a marker gene. What could be a marker gene for a bacteria? Can you tell me? A reporter gene or a marker gene for a bacteria.
So what is a marker gene?
Which tells you that if the gene is expressed, you will get to know. If your gene is there, it replicates and it expresses, you will get to know that your gene has expressed just by looking at the bacteria. Any idea? So I know that Salmonella typhimurium, let's say, it does not have resistance against antibiotic, penicillin for that, for example, okay? So it is not antibiotic resistant. Now I want to check whether this DNA recombination of plasmid and then putting that plasmid in the bacteria, this technique is working or not. So let me use the gene which provides the bacteria with antibiotic resistance. What say? Correct? So my gene of interest will be antibiotic resistance gene. So let us make a plasmid. What we all need for a plasmid? We need a desired gene, right? So let's say this is my gene of desire. This is antibiotic resistance gene, which was already not present in Salmonella. So we culture these bacteria in culture plates. So there is a media, agar media, which is food, on which these bacterial colonies grow like this. Sometimes they grow very close, but they completely fill the full plate. We know that this is bacterial colonies, they look whitish. So they are growing. Now, if you add 1 drop of antibiotic, what will happen? All the bacteria will die, right? If I put antibiotic on the plate, all the bacterial colonies will die away. Is it making sense, people? Yes, no. Am I audible?
Yes, sir.
Yeah, okay. So just to check this, now I took an antibiotic resistant gene with, what is this blast thing? This is origin of replication. I'm writing it ORI. So this is origin of replication. Then there is something else. Then there can be multiple origin of replication also. This is also another origin of replication. And this is the complete plasmid, let's say. Other things you don't have to worry about, but just know that in this plasmid, I have put my interested gene, which is antibiotic resistant gene. Now I put this plasmid into these bacteria. Then I cultured it, and I cultured them in an antibiotic environment. So now what will happen? If this technique has worked, if my gene of interest has been multiplied or cloned properly, and if it is expressed properly in the cell, protein has been made, then the bacteria now should not die in an antibiotic environment, correct?
Yes, sir.
So I will do a control and a test. In the control situation, I will put a normal plasmid or just an empty vector. We call it empty without our gene. So controls will have an empty plasmid without my gene of interest, which is antibiotic resistance genes. Then the test population will have the plasmid with my gene of interest. Okay. This is gene of interest. And in this case, you know it is antibiotic resistance gene. Now, both of these, I will put in both control and test plasmid. I will put in bacterial populations. How will I do that? I'll tell you in the next class. But now I will have two colonies, two culture plates where there will be two colonies. So controls are growing, but if I put antibiotic, what will I see? They will all die, right?
Sir.
Correct?
Yes, sir.
Yes, sir.
But if my gene of interest has worked,
So it will not die.
yes, if I add antibiotic while I'm growing these on culture plate, I will still get bacterial colonies, right? It will still have bacterial colonies. But whereas in this case, there will be no bacterial colonies that I will see. All will die. Correct? In this case, colonies survive. So what does it tell us? This tells us that my gene of interest has been
successfully recombinated, sir.
Yes, so I recombinated here only. So this recombination part happened here only. This is a recombinant DNA molecule that I made because my gene of interest was also genetically engineered in that, put in that. But this experiment tells us that my recombinant DNA has successfully been cloned and expressed in the bacteria, correct?
Sir.
Yes, sir.
Now, I can use the same thing to clone any gene of my interest. So with this antibiotic resistant gene, which is now a marker gene, I can just here, this part was still empty, right? So I can add here any other gene which is together with the marker gene. So let's say this is my gene X. It does something and I want to clone it. So I can do the same thing. And if I see again that bacteria are surviving, I'm sure that they are replicating the plasmid. And if they are replicating the plasmid, they are replicating antibiotic resistance gene. That's why they are surviving. So they must also be replicating my gene X because these two are linked together, correct? Under the origin of replication, correct?
Yes, sir.
So the number of copies that it will make for the antibiotic resistance gene, the same number of copies it will also make for my gene X. So this technique is working. This was first shown by two scientists, Stanley Cohen and Herbert Boyer. Very brilliant experiment in 1972. They isolated the antibiotic resistance gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. So they saw some colonies which could grow on antibiotics, and they realized that their plasmid have a gene already present inside, okay, in these Salmonella typhimurium, and they isolated, you can write down, they successfully for the first time isolated the antibiotic resistance gene. Okay. And how did they do this? This I'll take up in the next class. I'll stop here for any questions if you have. And your homework for the next class is you have to read about endonuclease. and exonuclease. Now I'm giving you a hint. If there is A's at the end, what does it mean?
It's an enzyme.
Yes, so you have to figure out what is this enzyme, what it does. Okay. In the next class, I will tell you about other tools. So these two are tools of making recombinant DNA. Okay.
Yes, sir.
Okay. Then we'll go to vectors that we can use for cloning. But is this part clear to all of you?
Yes, sir.
Very well. Any doubts, people? So please revise this because these are all going to be a little technical. Apart from biology, we are also using techniques, so it's going to be technical as well. So today I just give you an introduction and then we'll go deep into the DNA cloning and making recombinant DNA molecules in the next class. Please read about endonuclease and exonuclease before you come to the next class.